You know, when you break a bone, an X-ray gives you this really clean binary answer. You see that jagged white line on the film. The doctor points to it and says, well, there it is.
Right. Broken or not broken. It's simple.
Exactly. But when you step into the world of treating advanced cancers, that comforting clarity it completely vanishes. The picture goes totally dark. And today, we're diving into a different kind of diagnostic and therapeutic muddy water.
Yeah, we're looking at prostate cancer today.
Specifically, we're unpacking a groundbreaking 2026 paper from the journal Cell Death and Disease. And our mission for this deep dive is to explain exactly why these tumors are so notoriously resistant to modern immunotherapy.
And I mean, that resistance is one of the most frustrating puzzles in modern oncology right now, because we have these incredible breakthrough treatments like PD-1 inhibitors.
Which are supposed to be a game changer. Right?
Oh, absolutely. They're designed to take the brakes off the patient's own immune system so it can attack the cancer. But prostate cancer is what we call an immune cold tumor.
Cold meaning like inactive.
Yeah, exactly. You give the patient the immunotherapy and just nothing happens. The immune cells aren't just asleep, they're being actively, aggressively suppressed by the tumor itself.
So to sort of anchor this for everyone listening right now, imagine your immune system is a highly trained army, right? It's ready to attack a threat. But the prostate tumor has managed to build this specialized, almost invisible force field.
That's a really good way to visualize it.
And this force field, it doesn't just block your immune army, it actually captures your own soldiers and brainwashes them into protecting the tumor instead.
Right. The deceptive nature of it is what makes it so lethal. The tumor isn't just hiding in the dark. It's running a massive counterintelligence operation.
Wow.
And what this paper does, which is honestly so remarkable, is it provides the exact molecule-by-molecule blueprint of how the tumor constructs this wall.
Okay, well, let's unpack this, because I want to know what this force field is actually made of. Like, what are the specific cells standing guard?
So the primary guards are called MDSCs. That stands for myeloid-derived suppressor cells.
Myeloid-derived, so meaning they originate in the bone marrow, right?
Yeah, that's the origin point.
Yeah.
They are this highly heterogeneous population of immature immune cells.
Immature, meaning they haven't finished growing up.
Exactly. In a healthy body, myeloid cells are supposed to mature into your standard frontline defenders, things like macrophages or dendritic cells.
The ones that eat pathogens and present them to the rest of the immune system.
Right, the foot soldiers. But the tumor sends out these corrupting signals that hijack their development. It halts their maturation process entirely, keeping them in this immature, highly suppressive state.
That's insidious.
It really is. And the paper specifically highlights two distinct flavors of these cells. You have MDSCs, which are monocytic, and PMN MDSCs, which are polymorphonuclear.
Okay, monocytic and polymorphonuclear. I think the best mental image for these MDSCs, for you listening, is to view them as double agents.
I like that, yeah.
Like, they're born in the bone marrow, meant to be the good guys fighting infections, but the tumor intercepts their orders and corrupts them.
Yep, they go rogue.
But, how do we actually know they are there? The paper used some pretty heavy-duty tech to find them, starting with single-cell RNA sequencing.
Single-cell RNA sequencing is it's revolutionary for this kind of work. Historically, if you wanted to analyze a tumor, you'd grind up a piece of tissue and sequence the RNA of the whole sample at once.
Kind of like a biological smoothie.
Exactly like a smoothie. You're trying to guess the exact recipe just by tasting it. You know there are strawberries and bananas in there, but you don't know the exact proportions.
Right.
But single-cell sequencing lets researchers look at the smoothie and separate it back out into individual, intact strawberries, bananas, and blueberries. They analyze the gene expression of every single cell one by one.
That is wild. And they used a specific data set for this, GSC 141445, to map out the landscape of both healthy and cancerous prostate tissues. And the contrast was massive, wasn't it?
It was night and day. In a normal prostate, these double-agent MDSCs make up a tiny, tiny fraction of the cellular neighborhood, about 0.3%.
Okay, so barely there.
Barely there. But when they looked at the prostate cancer tissues, that number jumped to 2%.
Which doesn't sound like a lot on paper, 2%.
Right, but biologically, a 2% infiltration of highly suppressive cells is a massive structural change to that tissue environment. It's a huge defensive wall.
And they didn't just rely on the RNA sequencing to prove this, right? They backed it up with flow cytometry.
They did, yeah.
Which is such a cool technique. Flow cytometry is essentially making cells wear fluorescent vests and running them single file past a laser, right?
That's a perfect analogy. You use antibodies tagged with fluorescent dyes that stick to very specific proteins on the outside of the cells.
Like little glowing name tags.
Exactly. For these MDSCs, the researchers were looking for cells wearing very specific molecular vests. They had to be positive for the markers CD33 and CD11b.
CD33 and CD11b. Okay.
But they had to have very low or negative levels of a marker called HLA-DR. That specific combination is the undeniable signature of these suppressive double agents.
And the most alarming part to me isn't just that they found them hiding in the tumor tissue itself. When they ran the peripheral blood of prostate cancer patients through that same laser, they found a 4-fold increase of these MDSCs circulating in the bloodstream compared to healthy individuals.
Yeah, it's a systemic hijacking.
Hmm.
The tumor isn't just affecting its local neighborhood, It's broadcasting a recruitment signal system-wide.
Pulling these double agents straight from the bone marrow and circulating them throughout the entire body.
Exactly.
Okay, so let's do a quick recap of where we are so far. If you're following along, the defining characteristic of this cold prostate tumor is that it's swimming in these hijacked immune cells called MDSCs. Both the monocytic and polymorphonuclear types are dramatically elevated, building this localized and systemic wall against standard treatments.
That's the baseline reality of the disease, yeah.
So a question for you listening right now. Have you ever considered how a disease might not just hide from your body's defenses, but actively recruit them to act as its own personal security detail? It's terrifying.
It really changes how you view cancer. It's active, not passive.
Which naturally leads us to ask, how is the tumor actually pulling this off? Because these MDSCs don't just magically turn evil. There has to be a corrupting signal, like a mastermind inside the tumor pulling the strings.
There is. And the mastermind identified in this paper is a protein called RelB.
RelB. Okay, let's dig into that. What is RelB?
RelB is what we call a transcription factor. It's a critical component of the non-canonical NF-kappa B signaling pathway.
Now, NF-kappa B, that's famous in biology as a master regulator of inflammation and cell survival, right?
Oh, yeah, it's a huge deal. And the researchers found that RelD is highly expressed, meaning there's a ton of it in particularly aggressive androgen receptor-negative prostate cancer cells.
And they looked closely at 2 specific laboratory cell lines to prove this, known as PC3 and DU145.
Right, those are standard, very tough cell lines used in labs worldwide.
And the phrase androgen receptor negative is a crucial detail here. That means these are the really tough treatment-resistant cancer cells that just no longer respond to standard hormone deprivation therapies.
Exactly. They've evolved past needing those hormones.
So inside these aggressive cells, we have this RelB transcription factor. For anyone rusty on their high school biology, explain how a transcription factor actually does its job.
Sure. So think of the cell's nucleus as a massive locked library containing the DNA code for everything the cell could possibly do.
Okay, a giant library.
Right. A transcription factor is like a highly specialized librarian holding a very specific key. Its entire job is to travel into the nucleus, find one specific book, which is a gene, unlock it, and tell the cell to start reading it and manufacturing whatever that gene codes for.
Wow, okay. And the researchers actually mapped exactly which book RelB was opening in this case. They found it goes straight to the promoter region of a specific gene called CSF2.
Yes, CSF2.
And to prove it, they used a technique called chromatin immunoprecipitation, or ChIP.
ChIP, yeah. It's one of those elegant lab techniques that feels almost like magic when you explain it.
Tell us how it works.
So to figure out where RelB was going, researchers essentially flash freeze the cells using a chemical that acts like superglue.
Superglue on a molecular level.
Exactly. This glues the RelB protein permanently to whatever exact stretch of DNA it happens to be sitting on at that exact millisecond.
Oh, that's wild. Then they take molecular scissors, right? And chop the massive DNA strands into tiny pieces.
Right. And they use an antibody to fish out only the RelB proteins.
So they're pulling out the RelB and it drags the glued-on piece of DNA with it.
Exactly. Then they unglue it and they sequence that tiny piece of DNA to see what it says. And through this, they discovered 2 potential binding sites on the CSF2 gene.
But the data clearly showed that RelB overwhelmingly prefers binding to what they called binding site 2 or BS2.
Yes, BS2 is the target.
Okay, so our mastermind RelB travels to the nucleus and parks at binding site 2 on the CSF2 gene. Here's where it gets really interesting. What is the actual output of that gene?
Right, what does it make?
Yeah. When CSF2 is turned on, the factory starts pumping out a cytokine signaling chemical called GM-CSF, or granulocyte macrophage colony stimulating factor.
And that GM-CSF, That is the brainwashing serum we alluded to earlier.
Serum? Okay, break that down.
The tumor cell starts pumping this chemical out into the surrounding tissue. When that GM-CSF hits the receptors on the outside of those immature myeloid cells arriving from the bone marrow, it initiates this totally catastrophic cascade.
It flips a switch inside them.
It does. It activates a pathway inside the myeloid cells called STAT3. And STAT3 acts as the ultimate trigger here.
What does it trigger?
Two things. First, it causes those MDSCs to aggressively multiply, just rapidly thickening that force field. But second, it arms them to go on the offensive against your body's actual defenders.
By defenders, you mean the CD8 positive T cells?
Yes, the assassins of the immune system.
Right, the ones that are actually capable of recognizing and killing cancer cells.
Normally, yeah. But the MDSCs essentially paralyze them. A healthy CD8 T cell kills its targets by releasing highly toxic inflammatory cytokines, primarily IFN-gamma and TNF-alpha.
Plus their ammunition.
Exactly. But the MDSCs, driven by that STAT3 activation, they suppress the T cells. They physically stop them from producing those weapons.
Man. So without IFN-gamma and TNF-alpha, the T cells are just, they're soldiers standing on the battlefield with empty rifles. They can see the tumor right in front of them, but they can't do anything about it.
Right. The force field is complete.
So time for another quick summary check. RelB acts as the mastermind inside the tumor cell nucleus. It binds to the CSF2 gene at binding site 2 and mass produces the cytokine GM-CSF.
Yeah.
And then GM-CSF activates STAT3 in the MDSCs, causing them to multiply and paralyze our good CD8 T cells.
You got it. That's the mechanism.
So question for you listening. If you were a doctor looking at this factory, where would you strike? I mean, do you try to block the GM-CSF chemical floating around in the blood, or do you go straight for the RelB mastermind inside the cell?
It's a really tough strategic dilemma for drug developers.
It is. But wait, before we talk about treatments, I have to push back on the cellular logic here for a second.
Yeah, go for it.
Because proteins like RelB, they don't just get to hang around in the nucleus forever turning genes on, right?
Exactly.
The cell has a natural garbage disposal system for proteins that have done their job or are just misbehaving. They get chopped up.
Right. They do.
So how is RelB surviving long enough to build this massive force field in the first place?
That is such a great question, and you've hit on the central paradox of the whole paper. Under normal circumstances, RelB should be rapidly degraded.
Exactly.
What's fascinating here is how the tumor circumvents the cell's natural waste management system, which is formally called the ubiquitin proteasome system.
Ubiquitin proteasome. I always think of this system as the cell's wood chipper.
The wood chipper. The very accurate analogy, honestly. Normally, when a protein's time is up, the cell attaches a tiny molecular trash tag to it.
And that tag is a protein called ubiquitin.
Yes. For RelB specifically, these trash tags are designated to be attached at two very specific structural locations on the protein amino acids, known as lysine 295 and lysine 296.
Okay, so the trash tags get slapped onto lys 295 and lys 296, and then the cellular machinery sees the tags, drags RelB over to the proteasome, our wood chipper, and just grinds it into molecular dust.
That's the normal process. And the researchers proved this baseline reality by using a drug called MG132.
Right, MG132. That drug essentially unclogs the wood chipper, doesn't it?
It does. When researchers applied it to the cells, the proteasome stopped working and the levels of RelB skyrocketed. This proved that in a normal state, RelB is constantly being tagged and destroyed.
Okay, but in these aggressive prostate tumors, it's not being destroyed. The wood chipper is plugged in, it's running, but RelB is somehow avoiding it entirely.
Yes.
Something is stopping the trash tags from being attached to those two lysine spots in the first place.
And enter the paper's most novel discovery, a protein called ILF2.
ILF2, the ultimate molecular bodyguard.
The bodyguard, yes.
How does ILF2 actually protect RelB?
It's an incredibly physical mechanism based on 3D protein folding. You know, proteins aren't just flat lines of code. They fold into complex three-dimensional shapes.
Right, they have topography.
Exactly. ILF2 has a really high affinity for RelB, and it physically binds tightly to it at a very specific structural spot called arginine 176, or ARG176.
ARG176. And because of how the protein is folded in 3D space, ARG176 is structurally right next to our two trash tag spots, lys 295 and 296, right?
Yes, they are literal neighbors in 3D space. So when the ILF2 bodyguard stands on ARG176, its sheer physical bulk covers up the lysine spots.
Oh.
Yeah, the cellular garbage disposal system comes along with its ubiquitin trash tags, but it literally can't reach the attachment points. ILF2 is throwing itself over the target.
That is brilliant. RelB never gets tagged, so the wood chipper ignores it. RelB is stabilized, and it just stays in the nucleus, endlessly pumping out GM-CSF.
It's a perfect defense. And if we connect this back to the macro environment of the tumor, the researchers uncovered a vicious feed-forward loop here.
A loop? How so?
Well, the GM-CSF being pumped out of the cell doesn't just go out to brainwash the MDSCs. A portion of it actually binds to receptors on the very tumor cell that made it.
Wait, it feeds back into its own factory?
It does. That autocrine GM-CSF signal triggers a pathway that pulls even more RelB into the nucleus.
Oh, man.
So you have this self-sustaining engine. More RelB makes more GM-CSF. More GM-CSF pulls more RelB into the nucleus. And the whole time, the ILF2 bodyguard is standing there, shielding the RelB from destruction.
So putting all these pieces together for another quick recap, the tumor isn't just making a shield, it has engineered a perpetual motion machine of immunosuppression.
That's exactly what it is.
The RelB mastermind is stabilized by the ILF2 bodyguard, dodging the cell's natural garbage disposal. That creates an endless feedback loop of GM-CSF, and then that chemical goes out, recruits a massive army of double-agent MDSCs, which then thoroughly paralyze the T cells.
Right. And if you're trying to break a cycle like that, finding the exact right wedge to jam into the gears is critical. Because if you don't disrupt the loop itself, throwing standard therapies at the tumor is completely useless.
Which is a great question to throw to you listening right now. Think about the concept of feedback loops in your own life, like stress causing bad sleep, which then causes more stress. How critical is it to find the exact right wedge to break a cycle like that?
It's everything. You have to break the cycle.
So what does this all mean for the patient? We have this incredibly detailed, frankly terrifying map of the tumor's force field. How did the researchers use this map to design a weapon?
Well, this is where the basic science actually translates into clinical hope. They moved to in vivo testing using animal models. Specifically, they used mice implanted with RM1 prostate tumors.
And RM1 tumors are notorious for being highly aggressive and immune resistant.
It's really notorious, yeah.
And they decided to pull the wedge out of the feedback loop genetically first, right?
Yes. Through genetic engineering, they created models where they either knocked down the expression of the bodyguard, ILF2, or they knocked out the mastermind, RelB, entirely.
And what happens to a fortress when you remove the architect or the main line of defense?
The entire system collapses. In the mice lacking ILF2 or RelB, tumor growth just plummeted.
Wow.
When they looked at the tissue, the levels of those double-agent MDSCs had dropped dramatically. But the most profound finding was what happened to the immune system itself.
What happened?
With the force field dismantled, the good CD8 T cells came rushing back into the tumor microenvironment.
Because the GM-CSF factory was shut down, there were no MDSCs to paralyze the T cells anymore. The assassins got their weapons back.
Exactly. The T cells were fully active again. But obviously, we can't easily perform total genetic knockouts in human patients.
Right. We can't just delete genes in a clinic.
No, we need drugs that mimic that effect. So the researchers designed a therapeutic algorithm, a combination therapy, to test in the mice.
The grand finale of the paper, what was the exact drug combo they used?
They started with a specialized peptide inhibitor called SN52.
SN52. Okay, so a peptide inhibitor. This is a small chain of amino acids designed to block a specific interaction, right? What does SN52 do?
SN52 is targeted directly at RelB. It essentially intercepts RelB and physically prevents it from crossing the cellular membrane into the nucleus.
Oh, so if RelB can't get into the nucleus, it can't bind to the CSF2 gene, and the factory never even turns on.
Exactly. It stops the whole process upstream. But they didn't just use SN52. They combined it with a drug called nivolumab.
Nivolumab, that is an anti-PD-1 immune checkpoint blocker, right?
Yes, it's one of those breakthrough immunotherapy drugs we discussed at the very beginning.
Yeah.
The ones that traditionally fail in prostate cancer because the tumor is cold.
Right.
Its job is to block a specific molecular handshake that tumor cells use to tell T cells to stand down.
So let me make sure I'm getting this combo right. You have SN52, which shuts down the RelB factory and clears out the MDSC force field, turning the cold tumor hot.
Right.
And once the tumor is hot, nivolumab can actually do its job and supercharge the T cells to attack.
The synergy between the two was phenomenal. This dual combination achieved the absolute maximal anti-tumor effect in the models. It powerfully restored the T-cell effector function.
Meaning they measured massive spikes in those critical weapons, IFN gamma and TNF-alpha.
Exactly. The tumors shrank significantly faster and more consistently than with either drug alone.
That is incredible. Let's do one final recap of this therapy section. Disrupting the bodyguard ILF2 or the mastermind RelB genetically shrinks tumors and reduces MDSCs in mice.
Yep.
And the ultimate therapeutic combo they discovered is SM52, the RelB inhibitor, plus an anti-PD-1 drug like nivolumab. This dual approach dismantles the tumor shield while simultaneously supercharging the immune response.
That is the core takeaway.
So to you listening, this combination approach is like opening the heavy iron gates of the castle while simultaneously handing out upgraded armor and broadswords to the invading knights. Does this make you think differently about why a single magic bullet drug rarely cures complex, advanced cancers?
It really requires a systems-level approach to drug design. I mean, you are hacking a highly evolved communication network here. You can't just launch the offense. You have to surgically dismantle the defense first.
You really do. You know, looking back at this entire deep dive, I just have to marvel at the sheer elegance and honestly the deviousness of the biology here.
Devious is the right word.
Yeah. The tumor hasn't invented anything new. It didn't invent the RelB transcription factor or the GM-CSF cytokine. It simply hijacked the body's own natural inflammatory pathways and wound healing signals to build an impenetrable fortress.
That is the most profound takeaway for me. It's a form of biological judo. The tumor is using the body's own weight and momentum against it to survive.
It's a chilling thought. I mean, GM-CSF is fundamentally a good thing. It's a cytokine our bodies desperately need to rapidly stimulate immune responses during an acute infection, say, a severe pneumonia. We need it to keep us alive.
We absolutely do.
But here, in the murky waters of prostate cancer, the tumor has completely inverted its function to build a wall of suppression.
It leaves you with a lot to consider regarding how delicate our internal cellular ecosystems really are.
It really does. And as you go about your day listening to this, I want you to think about that balance. What other perfectly normal, helpful biological signals circulating in your bloodstream right now might be silently weaponized by diseases to ensure their own survival? And what other molecular bodyguards, like ILF2, are out there hiding in the data waiting to be discovered by the next generation of researchers?
That is the exact question driving the future of oncology.
Thanks for diving deep with us today. Keep questioning the obvious.